guys. We are the saviors of the soil. My name is Darwin Kim. I go to LaSalle High School in Pasadena.
I'm Kyle Herkins. I go to Laguna Beach High School down in Orange County.
I'm Jonathan Hung. I go to Shadyside Academy in Pittsburgh.
All right, so in abstract, as we all know, plants and microbes interact extensively through a symbiotic relationship. These communities of microorganisms, also known as microbes in the environment, live in and outside of plants. These microbes serve various functions such as decomposing and recycling old plant matter and providing plants with nitrogen and phosphorus, which is important for their chlorophyll, which is, of course, where photosynthesis takes place. So yeah, in this way, microbes indirectly affect us humans as well because we're the ones farming, appreciating the plant's beauty, and even eating the plants. So our scientific question is, how can we make plants and microbes safe from major metal pollutants such as zinc, copper, and lead? Our solution and approach would be to genetically engineer the microbes present in the soil, specifically Streptomyces griseus, which is a generic species found in almost all of the farming soil. We plan to edit the genome of these microbes and of these bacteria to make them more resistant to pollutants, using a gene that codes for resistance to these heavy metals, which would be purified from bacteria called C. metallidurans, which is often found in biofilm and in wastewater outside of industrial factories with high concentrations of heavy metals. So yeah, this question is significant because the symbiosis of plants and its microbes is currently endangered by certain threats such as climate change, soil degradation, and poor land management practices. Of course, we can't solve all of these problems, but we can try to eliminate at least one, therefore, and make a pretty big impact. And potential impacts is sustainable plants and microbes, and perhaps once our lab work is established, the global distribution of these genetically modified bacteria in the form of probiotics or maybe even in fertilizer. All right, so in concept, our question again is, how do we better protect plants and its microbes and its symbiotic relationship? And our mission is to preserve this harmony and address a problem, which is heavy metal pollution from runoffs and other things. So yeah, this topic is totally relevant to what we were learning because this is a cycle and it affects us humans as well. And it directly concerns the microbiome.
Thanks. Okay, so all the science stuff is great and all, but the important thing is, how does this affect everyone? How does this affect the global community? The soil that we're looking into is the kind of soil that would grow on farms, or the kind of soil that would grow our food. So if this soil has these pollutants in them, those pollutants can eventually find their way into our foods and eventually us. And personally, I don't really want to eat polluted food. So by seeing what we can do about the pollutants in the soil, we can potentially make our food even safer to consume. Also, by looking around in the soil microbiome, we can possibly find other problems that could get into our food and attempt to solve those as well. Next slide. Thank you. Pollutants in soil is not a very new topic that people have been looking into. There are many studies that have been looking into how the soil is affected by these pollutants. However, very few of these studies are actually looking at how the soil microbiome is affected by and affects these pollutants. So that's what makes our study very special. We are actually taking it down to that very small scale to see how things down there affect the big picture with the soil. Yes.
All right, so the first step in our team's approach would to be we need to find a bacteria that has desirable traits that we'd want in the plant microbes. So the one we found was C. metallidurans, and this bacteria is resistant to heavy metals and is able to ingest these heavy metals and make a non-phototoxic byproduct, which just means these byproducts won't harm the plants. So after we extract the DNA, we're going to try to cut out the DNA responsible for these traits and replicate them using PCR. So just PCR, the first step, you would heat up the mixture to break the double bond, the double helix, I mean, and then you would cool off the mixture and add primers so that they can attach and add nucleotides. And then you would heat the mixture up again, and the nucleotides would add to the primers and extend, and then you would have more DNA. And then you just repeat these steps until you have a bunch of replicated DNA. So now that we have our PCR product, we want to get it into the plasmid of plant microbes. So we would take the plasmid out and cut it with restriction enzyme, and then we would try to ligate the plasmid in our PCR product. And then after we have these plasmids containing this DNA code, we would heat shock it back into the plant microbes so that hopefully they can express these traits. So to check to see if these plant microbes actually took up the plasmid, we'll grow them in a dish with heavy metals present so that only the plant microbes with the heavy metal resistance will be able to survive. And then to see if they have the other trait, which is we want them to be able to ingest these heavy metals and also create a non-toxic byproduct. So to check that, we'll look at these bacteria under a fluorescence microscope and see if they're actually doing this process. So our greatest challenge is finding bacteria with these traits so that we can put them into plant microbes. So for our example, it's C. metallidurans. And this bacteria is really important because it not only is resistant to these heavy metals, but it's also able to convert these heavy metals and toxins into a byproduct where the plants aren't able to consume it. So we don't want to be eating plants with these toxins in it. So to sum it up, we're looking for bacteria that have these desirable traits that we'd want to put into the plant microbes. We're going to take the DNA out and replicate it using PCR, and then we're going to ligate it into the plasmids of plant microbes, and hopefully they'll display these traits.
Okay, so these are our team members here, and we are done presenting. So thanks for listening. So we have time for some questions. So if you have a question, go ahead and ask. No questions?
All right.
Okay.
So one issue is your plan is to take microbes that are universally available in plant soil and make them able to convert these metals into a non-toxic byproduct.
Yes.
Right? So in this case, first off, let me compliment you on the level of detail in terms of being able to actually plan this genetic strategy to basically transfer these genes and see if you can develop this new property in your resulting microbes. I think that's actually a very, very good detailed explanation of how you would approach that. But the question would be then, after you find this bacteria and you spread them out into the wild, how are you going to make sure that they thrive? Right?
So what we're planning to do, with the bacteria, we're going to start small. We're going to keep it to like maybe farm areas. We're not going to try to expand it all around the world. And we'll have to have a pretty good sample. Otherwise, as you said, if we only put in only a few of the modified bacteria, they might die, and then we'd have to start back from square one. So, yeah, so we'd start small, we'd see how it goes, and we'd make modifications as we go from there.
And maybe we could add on some sort of like genetic code or like advantage to give them a step up over the like wild or natural microbes so they would thrive.
On a farm like
Yeah.
pesticide resistance or something like that.
Yeah.
You have to look at how that affects microbes. But
Yeah.
interesting ideas.
Okay. Yes?
So you chose fluorescence microscopy from the 3 techniques you learned.
Yeah.
In terms of the proposal you have, which I agree with Mike, is quite nice, what is the key insight you would gain from using that technique about your proposal with respect to either visualization of these microbes or the actual output of the
Right. So from the various techniques that we learned this week, obviously it's a biological sample, so we don't want to kill it using electrons. But I think since we're dealing with bacteria here, we only need to look at the cellular level, and we need to make sure if it's producing the non-toxic byproducts. So any typical light microscope would do, and we thought that fluorescence would do the job.
So I guess I'll take it further. So what would you hope to see from the fluorescence, right? Because the images that you showed, my understanding would be you would have to fluorescently tag some element that showed that you're basically insertion of this DNA has occurred. Right? So you know that you've manipulated these bacteria. But then how do you know that they actually are doing what you think they're doing?
Right.
Using microscopy.
Right.
There are other readouts that you could use.
Right. So under the microscope, in the presence of these heavy metals in aqueous solutions, to make sure that it worked and that the bacteria have the desired trait, we would be able to see the byproduct, which is solid metals and solid copper, solid lead, things like that. So, which is of course, which is hard for plants to digest. Yeah, so that's what we'd be looking for solid metals. Yeah, I think we would be able to see that under a microscope.
Is there one more question?
Students?
So if we don't have any more questions, then we're done here. So thanks for listening again.